doctora Jennifer Manilay. La profesora Jennifer Manilay posee un doctorado en biología de la transplantación y desarrollo de linfocitos. ¿Dije bien? Linfocitos. Ando con el doctor Michael y se me olvida el castellano. De la Universidad de Harvard. Es profesora titular en la Universidad de California, Merced, con más de 20 años de experiencia en investigación e inmunología, biología de células madre y osteoinmunología. Así que les pido que recibamos con un fuerte aplauso a la doctora Jennifer Manilay.
Buenos días. Buenos días. Gracias, gracias. Me llamo Jennifer Manilay. Soy profesora en Universidad de California en Merced y no hablo español. Mi español no es bien. So I will give my presentation in English. Please use the translation in Zoom if you prefer. So first, thank you to the organizers for inviting me here today. It's my first time in Colombia, and it's my first time speaking to audience that doesn't speak English, speaks Spanish. And so I'm a little nervous, soy nerviosa. So before I begin, I just want to thank everybody for this opportunity. I'm excited to meet with all the students and the faculty to talk about possible opportunities and collaborations between our universities. So thank you. Today, I'm going to talk about my research. In this talk, I'm not going to talk about viruses. No viruses. Okay. I'm going to talk about my research in immunology and my story. So similar to Dr. Ceballos, my journey follows my interests. And so my questions change over time. And it's a lot of fun. Okay. So before I start, I want to say again, it's my first time in Colombia. Y ayer aprendí una palabra nueva, bacana. So if you don't understand any of my talk, my goal is to show you mis investigaciones son bacano. Okay. Okay, so me llamo Jennifer Manilay. I'm a professor in UC Merced. I've been there since 2005. So I'm one of the first faculty at UC Merced. I'm Filipina American, and I'm the first American born in my family. So I have a lot of firsts in my history. So during this symposium, there's many opportunities for you to meet me, and please introduce yourself to me. I really want to talk with you. So I have 3 talks. The first one is happening now, right? The second one will happen this afternoon, and I hope you will all come because we will have some activities during that time. And then tomorrow I will give a workshop on how students may get some research and also apply for graduate studies at University of California, Merced. Okay, so this is an outline of my talk. And so first I'm going to start talking about the immune system and introduce you to immune system cells and the bone marrow. And I will tell you about a concept called the bone marrow niche and how the bone marrow niche supports blood cell development. And then I'm going to tell you 2 research stories about my studies in osteoimmunology, which is the interaction between the immune system and the skeletal system, and how these studies can have implications for diseases that impact immune cell homeostasis, inflammation, and immunotherapy. Okay, so first, I will begin with this slide. I am a developmental immunologist, and in developmental biology, stem cells are a focus. So my research projects connect immunology, hematology, developmental biology, and stem cell biology. So as developmental biologists, we define a stem cell as its ability to self-renew, and also to produce another copy of itself, as well as its ability to differentiate and produce a mature cell of a defined lineage. So in my research, we study pluripotent hematopoietic stem cells. These are the cells from which all the immune cells develop. And so this is the diagram of all of the immune cells that come from this single pluripotent hematopoietic stem cell. So today I'm going to tell you some stories about my research that focused on B cells, which are your antibody-producing cells, as well as erythroblasts, which produce red blood cells. Okay, in mammals, the hematopoietic stem cells reside primarily in the bone marrow and can be found in different bone types, including your femur, tibia, ribs, sternum, arm, and even your skull. And within the bone marrow, there's been much work and research from many groups that have shown that the bone marrow microenvironment is dynamic. And there are interactions between hematopoietic cells, so blood cells, and non-hematopoietic cells. So today I'm going to focus on the bone marrow stromal cells that have been shown to support B-cell lymphopoiesis. These are the mesenchymal stem cells, osteoblasts, and osteocytes. So this is a cartoon that depicts the different locations of the stromal cells within the bone marrow relative to each other. And it shows where the hematopoietic stem cells, shown in green, are located. So mesenchymal stem cells are the precursors of osteoblasts here. And osteoblasts are the bone-building cells in your body. The osteoblasts reside in a niche called the endosteal niche, and they can differentiate into osteocytes. These are the osteocytes. So osteocytes are the mineralized, hard cells of your bone that make your bone strong and hard. And they're the cells that can sense mechanical loading inside your bones. Okay. In addition to bone cell development, mesenchymal stem cells, osteoblasts, and osteocytes regulate hematopoiesis and immune cell development in the marrow by direct cell-to-cell interactions through the secretion of growth factors, which I'll tell you about these, CXCL12 and stem cell factor. And it's thought that migration of immune cells to different stromal cells in the marrow is important for their normal hematopoiesis. So all of this background is related to my research questions. And so I'm interested in basic research in understanding cell fate decisions and differentiation in the immune system. So some of the questions I ask are, what directs a blood stem cell to become a specific cell in the immune system? How does the stem cell know that you need to produce more B cells instead of more macrophages or more blood cells? Okay. How are the immune cells activated? How do they recognize what is self and what is foreign? I'm getting older. And I am interested now in how immune cells are affected as we age. And this has led to my questions on how does bone disease affect blood cell, stem cell development and function. So today, I'll focus on my research in osteoimmunology, which is an interdisciplinary field. And I'll show you some of the work that we do in my lab. So in my lab, we use a lot of cellular and molecular techniques, such as cell culture, PCR, microscopy. I use a lot of flow cytometry, which is very important for immunology research. And my model organism is mice. So today I'm going to focus on my recent work in osteoimmunology. And to give you some context on how I fell into this work, I'll start with my first story on sclerostin. Okay, so you should be able to read this slide. Okay, so osteoporosis is a disease that is often acquired with aging, and it results in weak and brittle bones that can lead to risk of bone fracture. Osteoporosis is a worldwide problem and a major health issue for the elderly. Most medications to control osteoporosis focus on inhibiting bone resorption by osteoclasts, but only recently new drugs that focus on bone building have become available. So recently, there's clinical trials for a new osteoporosis drug called romosozumab. And it is an antibody against a protein called sclerostin. Sclerostin is the protein that promotes osteoblast differentiation to osteocyte, and it's very important for regulating bone building. So when you block sclerostin with an antibody, it enhances bone building. And you can see this here. These are scans of the bone of mice that are treated with the sclerostin antibody. And this shows that there's more density inside the bone marrow cavity compared to saline-treated controls. So this has been tested now in humans, and it's very successful for patients that have osteoporosis. However, and it's very exciting because it's the first new drug that focuses on bone building cells. All the old drugs focused on osteoclasts and have a lot of side effects. So this is very exciting in the skeletal biology field, osteoporosis field. However, sclerostin is also very important for regulating the hematopoietic system. So let me tell you a little bit about sclerostin. So sclerostin is a Wnt antagonist. If you've taken cell biology, you learn about hormones that can activate cells. And Wnt is a secreted glycoprotein that is produced mainly by osteocytes. So the way it works is it binds to the Wnt signaling co-receptors, LRP4, 5, and 6, and its job is to block Wnt signaling. And because of its role in bone homeostasis, it's really, really popular to study in the skeletal field. So how it works in the bone is that when Wnt signaling is important throughout development, from the mesenchymal stem cell all the way to the osteocyte. And so as the bones are developing, there is Wnt activation required for development. And then to get to this critical stage, osteoblast to osteocyte, you need to downregulate Wnt signaling. And that's where sclerostin comes in. So as I mentioned, anti-sclerostin therapies are a novel emerging treatment for osteoporosis. And it works by inhibiting bone building osteoblasts. And in humans that have a mutation in the SOST gene, they have a disease called scleriosteosis, which is over mineralization of the bone. And in Van Buchem disease, the bones keep growing and growing. And you can see this in the skulls and the face bones of these patients. They have deformities. So these diseases are characterized by excessive thickening of the bone tissue. And this is the strategy to use this sclerostin as an application for osteoporosis. However, Wnt signaling is not unique to the skeletal system. It's also involved in hematopoiesis. And so Wnt signaling in hematopoietic cells works the same way. And Wnt3A and Wnt5A, which are ligands for HSCs, are important for their self-renewal. Wnt10B is a primary molecule involved in osteoblast fate decisions. The frizzled receptors can bind to multiple Wnt proteins. And the role of Wnt antagonists like sclerostin has been explored in the hematopoietic niche. But it's very rare. I think maybe I'm the only one, right, that tries to look at hematopoiesis and the skeletal system in the same way. So when SOST is present, it turns off the Wnt signaling pathway. Okay, so as I mentioned to you, your hematopoietic cells and your immune cells develop inside your bone marrow. And so if you affect the bones, my hypothesis was that you can also affect the immune system and hematopoietic system. And so a long time ago, one of my doctorate students started this work as part of his thesis, Corey Kane, and in 2012, we started publishing on this work. Okay? And so just to quickly summarize, these are some papers. They're all available and published in open access, so you can read them later if you so what we found first is that our first study showed that SOS deletion can affect immune cell development in a bad way. Okay. Our first study identified increased apoptosis of B cells in the bone marrow of SOS knockout mice, but we did not detect any effects on Wnt signaling in the B cells. Instead, we found that the B cell defect in the SOS knockout caused levels of a B cell growth factor, CXCL12, to be decreased in the SOS knockout stromal cells. So the bone cells could not produce the growth factor for B cells. So this led us to conclude that the bone marrow microenvironment was altered due to the changes in the bone structure in the knockout mice. We then followed up this study with a series of conditional SOS knockout mice where we could knock out the SOS gene just in mesenchymal stem cells or just in osteoblasts or just in osteocytes. And what was surprising in this study was that it was the SOS gene that was in the mesenchymal stem cells that contributed to the B cell bone loss. But if you knocked it out in the osteoblasts and the osteocytes, there was no effect. So that was very surprising. Okay. Then in these two studies, our first two studies, we focused on younger mice that were 12 to 16 weeks of age, which is about maybe your age, about 20-year-old human. Okay? So in our most recent paper, published 2021, we looked at older mice. And what we found was that in the sclerostin knockout mice, in addition to having more hematopoietic defects with age, they also showed evidence of inflammation in the bone marrow. And inflammation in the bone marrow can indicate that there is accelerated aging happening in the bone of these mice. Okay, so what this means, or what I think this means, right, is that people that receive sclerostin antibody treatments for osteoporosis might have side effects on their hematopoietic system. And so I'm trying to get some collaborators now to get some patient samples to investigate this. It's a little bit challenging because the drug companies don't want to collaborate with me because my data in mice suggests that something may be happening with their drug. And I've asked the drug companies and they have declined my request. So, so I need some new collaborators, right? But in any case, every year when I go to the bone and mineral research meeting, my research generates a lot of interest from the basic scientists. And so I guess the moral of the story is I didn't go into skeletal research looking for a problem. I saw bone that didn't have any bone marrow, and I was interested in the immune system and what was happening with the immune system in those mice. And in the course of working with students and chasing this curiosity, I may have uncovered some evidence that has applications to humans in clinical trials. So this is ongoing research. Okay, so next I want to tell you my second story. And this is newer work with another knockout mouse called the VHL knockout mouse. And this work is our most recently published work. There's 2 QR codes here if you would like to look up the papers. The first one was published last year in Frontiers of Immunology, and the first author is Betsabel Chicana, who is was a PhD student in my lab. And then the second one is under review, and the lead author is Jana Emery, who is was a master's student in my lab. So I'm just going to give you a little bit of a summary of both of their projects, because each of these projects took 5 years to take, and I only have 45 minutes. No, I only have now 23 minutes to finish the story. Okay, so VHL. VHL, what is it? Right? It is a gene that is involved in the sensing of hypoxia in cells, so oxygen levels in cells. It encodes for a ubiquitin ligase of hypoxia-inducible factor, which is this protein here. And in normal oxygen conditions, this HIF protein is inactive. However, under hypoxia or low oxygen conditions, the VHL protein is not able to ubiquitinate HIF, and therefore it's not degraded like in the normal situation. So in hypoxia, the HIF proteins will then translocate to the nucleus, where they bind with other proteins to start transcribing HIF target genes, or hypoxia-inducible factor target genes. So we use the same strategy. We studied a VHL knockout mouse that doesn't have produced this protein in bone cells. And so in that mouse, the hypoxia signaling pathway is always active. Okay. Okay, so why is this important? Right? So HIF stabilizers are another target for osteoporosis. So there's been a lot of studies in mice where the VHL and HIF signaling pathway has been targeted in the skeletal system. And when you inactivate it, it starts to build more bones. So here's another drug pathway. So we were curious to see how immune cell development was affected in the VHL knockout mice. So here you can see these are scans of the bone of a control mouse, and the black spots here show you the spaces where the bone marrow is. And in the VHL knockout mouse, there's hardly any bone marrow, like none, right? And you can see this too. This is the thickness of, this is a cross section of the long bones, and it's much thicker in the VHL knockout mice. Okay, so our goal was to understand how changes in bone homeostasis driven by deletion of VHL in osteocytes, which are DMP1 expressing cells, affect B cell development in the bone marrow. And so we examined the niche cells and cytokine levels. We looked at bone marrow organization, and we also examined the level of oxygen in the bone. Okay, so this is a busy slide, a lot of data, so I will try to go slow. Sorry, translators, if there's a lot of words here that don't translate well. So, okay, in this, first, I'll tell you the conclusion first. We saw defects in B cell development, very severe in the VHL knockout mice. So here in this graph, we see a significant decrease in the cellularity of the bone marrow. We saw a decrease in the B cells, both in number and frequency, shown in yellow. And we wanted to see exactly where during B cell development this block was occurring. So we used flow cytometry, and we used a strategy that was described by Richard Hardy in 1991, where he named the fractions of B cell development fraction A to fraction F. So least mature to most mature. And so what you can see in this graph is that in red, which is the knockout mice, the number of B cells in all of the mice is reduced compared to the controls. And we also did transplants of wild type bone marrow into the VHL knockout mice to show that it was really something with the microenvironment in the VHL knockout mice that was affecting the wild type bone marrow. So normal bone marrow put in a VHL knockout bone does not produce B cells. Okay, so we wanted to understand what's going on in the bones of the VHL knockout mice. And so again, we use flow cytometry. These are some plots here. If you have not seen these kinds of plots before, on the top is an apoptosis assay where we stain for annexin 5 and 7-AAD. Annexin 5 stains for a marker on the cell membrane that flips to the outside of the cell when it's dying and going under apoptosis, and 7-AAD binds to DNA. And so if it can bind to DNA, that means the cell is permeable and also not healthy and undergoing apoptosis. So you can quantify the number of cells going through apoptosis, through late apoptosis and early apoptosis with this assay. So what we found is that there's increased apoptosis in the knockout mice starting at fraction B. We also looked at cell proliferation using another flow assay. So Ki67 is a nuclear antigen that is activated when a cell is in an active cell cycle, and DAPI stains DNA. So here in the control, there's a lot of DNA staining. This is an actively dividing cell, whereas in the knockout, you see the peak is very small. And so this is indicative of decreased proliferation. So we have increased cell death in the B cells, and we also have decreased proliferation in the B cells in the knockout mice. So why? Right? So what's driving this cell death and reduced proliferation? So next we measured growth factors. We measured cytokines. So earlier in the talk, I mentioned to you that there are two growth factors for B cell development in the bone marrow, CXCL12 and stem cell factor. And using a multiplex assay that stains for many different cytokines, we found that CXCL12, which is a key B cell growth factor, was significantly reduced, but stem cell factor did not seem to be different. Okay. In addition, this assay also showed an increase in erythropoietin, EPO. And I'm going to come back to this towards the end of the talk. So erythropoietin is very important for red blood cell development. But importantly, what this high levels of EPO indicates that there is hypoxia or low oxygen sensing in the bone marrow of these mice. So to follow up on the hypoxia, we used another assay called PIM staining, where PIM stands for pimonidazole, right? And the way that it works is that it is a hypoxia cell marker. So PIM will stain to cells that are actively experiencing hypoxia. So in this graph, the PIM staining on the knockouts is shown in red. And so first, as a control, we stained LSKs cells, which include hematopoietic stem cells and hematopoietic progenitor cells. And they have been shown in the past to have to reside in a hypoxic environment. So you see a higher staining of PIM in both the controls and the knockouts at 10 weeks of age. But in the older knockout mice, this hypoxia marker is increased. And then we continued to drill down, and we looked at the different fractions of B cell development. And we saw that at the 6 months age, the fraction A cells were expressing high levels of PIM, which indicates that there is hypoxia happening in the bone marrow of these mice. Okay, so now we are following up on the hypoxia studies. And so in collaboration with a bioengineer, Professor Joel Spencer at UC Merced, we are doing ex vivo and in vivo imaging. And this is just an example of some of the studies we are doing. So in this slide, what we have done is we've transplanted cells from mice where all of the B cells express a green fluorescence protein into control mice or VHL knockout mice, and then examined the location of the cells after transplant. So here in the top, you can see first, here's the bone of the control mice, and the red line here shows you the bone marrow, and it's throughout the whole bone. Whereas here in the knockout mice, you can see the space is much smaller. And then when we look for the green cells, the B cells in these control mice, we can see that they are very close to the growth plate and the metaphysis of the bone. So here are all the green cells. Whereas in the knockout mouse, all these blue represents a bone, and the B cells are moved more towards the diaphysis of the bone. So this shows and suggests that the different locations of the B cells is affecting their development. Okay, so if you are a patient receiving a drug to treat your osteoporosis, you might also, that targets the HIF pathway, you might also be affecting your B cells. Okay, so following on the theme of chase the data and your curiosity, my student Jana Emery joined the lab, and her background was more in physiology, and she was very interested in the increase in the EPO levels, the erythropoietin levels, because she wanted to know more about the hormone levels and how red blood cell development was affected. I don't know anything about red blood cell development. And so I told her if she wanted to study this, that she needed to help me learn. And so we learned together the different ways to analyze red blood cell development. And so the theme of her master's thesis was to examine how VHL knockout in the skeletal cells affects erythropoiesis. And so just to remind you of the stem cell graph I showed you earlier, the hematopoietic stem cells can differentiate into all of the cells of your immune system, as well as the red blood cells. And the precursor of red blood cells is called the MEP, which stands for megakaryocyte erythrocyte progenitors. So we'll come back to this in a moment. Okay, so what Jana taught me was that erythroblasts also develop in a specialized niche in the bone marrow called erythroblastic islands. And during the development of red blood cells, there are special macrophages, which are immune system cells that kind of nurse the red blood cells just through their different stages of maturation, which are illustrated here. And these stages of RBC or red blood cell development have very long names: proerythroblasts, basophilic erythroblasts, polychromatophilic erythroblasts, and orthochromatophilic erythroblasts. I don't know how you say these in Spanish, right? But these are the stages, and we can detect these different stages using flow cytometry. Okay, so what Jana found is that using flow cytometry, she has found evidence for accelerated steady-state erythropoiesis in the VHL knockout mice. So here again are examples of some flow cytometry plots that analyze red blood cell development. And so in contrast to the B lymphocyte development, which was decreased, the red blood cell development in the VHL knockout mice was significantly enhanced. So you can see a much higher level, this is shown in here red, of the red blood cell development as the mice age from 3 weeks all the way to 24 weeks. And so Jana also looked at complete blood cell counts in these mice. And so this is the test that is similar when you go to the doctor and you give some of your blood, and then they run the differentials and the red blood cells, and they count the white blood cells. We did that for the mice. And so consistent with our flow cytometry data, there's increased red blood cell counts in the VHL knockout mice. And this correlates with an increase in hemoglobin and the hematocrit, which are measurements of how well your red blood cells can bind to oxygen. This hemoglobin is the protein that carries oxygen. Okay, so then we asked, okay, what's going on in the mice to drive this red blood cell differentiation? And so we started using more omics technology. And so we have now done some bulk RNA sequencing of the MEPs, the mega erythrocyte progenitor cells from the VHL knockout mice. So we isolate these cells by flow cytometry, so they're very pure. We can isolate the RNA, and then we send it to a core facility for sequencing. And what bulk RNA sequencing permits you to find is all of the genes that are expressed in your cell type, and then to compare to your experimental samples. And so we found that in the VHL knockout mice, which are these two columns on the right, that there was very upregulated expression of genes involved in hypoxia, also genes that are involved in erythrocyte development, right? And also genes that are involved in glucose metabolism, and 419 other genes that we're not sure what they do yet, right? So we have lots of questions to investigate here. Okay, so what I hope that I have told you today, I've convinced you that crosstalk between the skeletal and immune systems is important for the maintenance of B lymphocytes and bone marrow in adult mice, that skewing of bone homeostasis towards bone building can adversely affect B lymphopoiesis, as well as augment erythropoiesis over time. And so the evidence for this statement comes from our studies from sclerostin antibody depletion and sclerostin knockout mice, as well as the VHL knockout mice, which stabilize hypoxia response pathways. And so taken together, these findings have implications on immune and hematological status of patients receiving new anabolic therapies for osteoporosis. Okay. So just to briefly summarize what's going on in my lab now, we have some current studies going on where we're trying to create better mouse models to investigate molecular mechanisms. So we have been successful in creating a osteolineage specific knockout mouse for that we can use to knock out any genes in osteoblasts using a tamoxifen inducible Cree. And so we're just starting this experiment now to produce conditional inducible VHL knockout mice. we are proposing to use single cell RNA sequencing to get more detailed analysis of skeletal cell types in skeletal progenitors and hematopoiesis. And so this is just an example of some of the single cell RNA sequencing that we've done with my collaborator, Dr. Lutz at Lawrence Livermore Labs. I'm also trying to get new funding to follow up on the sclerostin project in aged mice. So I'm proposing to get some aged mice that are 18 months or older, which is equivalent to a 50 to 65-year-old human, which are the age of patients that usually start taking osteoporosis drugs. And so I'm hoping to get some funding to analyze the molecular and cellular changes in the mice that are equivalent to older humans. And still collaborate with doctors that will help me study whether this phenomena is happening in human patients. Okay, and then a totally different project that is happening, which I'm going to tell you more about this afternoon, is a study where we just started this summer looking at the long-term effects of pesticide exposure on the bone marrow. And so I'll show you this slide again if you come back this afternoon. So more information in my next presentation. Okay, so to finish, I just want to acknowledge these are the students that were involved in this project. In my lab, I utilize the talent of a lot of undergraduate students. They come to my lab with no experience and they leave my lab as co-authors on published papers. And so here is, and then I have graduate students too. So here is Betsabel and Jana, who are the co-authors on the papers I told you on the VHL assay. And these are a couple of our undergrads who graduated last year. And also this work involves a lot of collaboration with scientists from different disciplines. And so I find it very fascinating to work with people who don't have the same expertise as me because our conversations are very rich. And it's really nice to hang out with interesting and smart friends, right? We start to chase questions that are very unique in this way. And I'll tell you more about that this afternoon. So thank you, and I'll gladly take any questions.
Thank you. Gracias, doctor Jennifer. Manilay, ¿qué preguntas tenemos en el auditorio, por favor? ¿Quiénes desean formular pregunta? Allá atrás. María del Mar. Mira, buen ejercicio ahí atrás. La niña, ¿eres tú? Sí. Ahí van con el micrófono. Gracias por la presencia de ustedes y también a aquellos que están de manera virtual a través del sistema de Zoom, atendiendo este importante simposio. Adelante, nombre por favor y nos formules la pregunta.
Oh, okay, I don't need this then. Gracias.
My question is, this is like a change of the treatments for osteoporosis because you discovered that the effect is really affect for the red blood cells. So this will change the treatment for these kinds of disease.
So that's what I think. And but my findings are in mice. And so we need to confirm that these are happening in humans. It's a simple question, but it's not easy to get that evidence scientifically in the lab. And that's my challenge right now. So I don't know for sure if what I find in the mice is happening in the human patients.
And you can test it with the cells, not with the human patient, but with the cells.
If I can get some samples of the blood and maybe bone marrow from the human patients, then I can do similar studies to confirm changes. But it might take, there's some caveats. So it might take a long time to see the changes. And usually in clinical trials, they only look in one time point and don't follow up, later. And so it's a little bit challenging. It's also really hard to get people to just donate their bone marrow because you have to put a drill in your hip to get the bone marrow out. So it's hard to get that from humans. Plus, you need to have all the permissions and regulations. So I'm trying to find a partner who's in the clinical trials right now.
Thank you so much.
Mhm. Oh, and then follow up. But I think we could do similar studies in larger animals, right? Which are a little bit closer to humans. But, I'm impatient, so I want to go from mice to humans, but we could probably look at some of the larger animals along the way. Otro?
Sí. Acá y luego allí. Acá. Acá. Nos das tu nombre y luego seguimos con la señorita que está ahí. Tendríamos 3. ¿Alguien más? ¿Un cuarto que desee preguntar? Bueno, entonces, estas 2 preguntas y vamos.
¿En español o en inglés? Oh, gracias.
My name is Joshua, and my question was kind of related to this. Like, is there a way, or it would be easy to get to work with veterinarians, so these treatments are applied to dogs and cats, so that way you can get an easier way to work later with humans, since, let's be honest, people don't see dogs and cats as the same as humans.
Yeah.
So is there an easy way to work with medical treatments in them first? Since I have seen that a lot of pets in today have osteoporosis and problems like that. And so what kind of progress have you done with that?
So you're my new friend. I don't have any veterinarian collaborators. We don't have a veterinary school at UC Merced. And so I would love to collaborate with you. I don't know anything about osteoporosis in dogs or cats and the prevalence of it. So let's work together, please, because I think it's really important and it will be helpful for our dogs and cats too. Thank you. Mhm.
Hi, my name is Laura.
Oh, in English.
And I don't know if I got everything correct, because like my scientific language is not that good, but if you were saying that the stem cells can help, like they renew, and they can help to increase the blood cells, that also could help the bones to like be stronger.
Mhm.
So if it is possible to like stimulate the stem cells for them to renew, and then you could take them. I don't know if this is possible to take stem cells from younger mice and place them on an older one.
Yeah.
So if you could like investigate that, and you could do the same with humans, you could cure the osteoporosis itself.
Yes, that's a great idea. So there have been some studies that have taken stem cells, hematopoietic stem cells, the blood stem cells from young mice, and put them into older mice and showed that they age, right? Skeletal stem cells are a little bit different and they're harder to isolate. They're still controversial how to isolate them, and they're not as easily transplantable. So my strategy is thinking to manipulate the blood stem cells to help the skeletal cells because they communicate with each other. But your idea to use the younger stem cells is already being tested as a way to reverse aging and some other ideas, but it's still being worked on. It's still an area of active research.
And is it possible to make it like not, I don't know if it's a thing, but if you could make it like synthetic, like you could take the stem cells and turn them into like potentiated ones.
Yeah.
In our laboratory, and place them. I mean, not meaning a younger, like a younger being,
Mhm.
but just use it like in the laboratory.
Yes, yes, absolutely. So in stem cell research, there is a lot of new research that is trying to take cells from anybody and you may induce them to be pluripotent stem cells. You might have heard of IPS cells, induced pluripotent stem cells. So I haven't worked on that in my lab, but that's going on in a lot of different labs. Actually, right now we're trying to get some grant funding to get a lab on campus to start working on human IPS cells. And so if we get that funding, then that will help me move in that direction. But yeah, you're good questions, very good questions. Thank you. Mhm.
Hi, I am Fernando
Hi, Fernando.
from the Universidad Libre. First of all, it's a marvelous presentation. Congratulations.
Thank you.
Beautiful presentation. And I have some basic questions. One of them is what hypoxia, what the level of oxygen on hypoxia cells that you're studying?
So we haven't, the assays that I have done so far, they're just a proxy for hypoxia. My collaborator, Joel Spencer, the bioengineer, he has some oxygen probes that we're trying to test in vivo that can really measure the exact levels of oxygen in different parts of the bone marrow. So he has shown in the past that in his previous studies that the hematopoietic stem cells are in a hypoxic environment of the bone marrow. And so we're trying to translate that technology now to our mice. And it's a little challenging because our bones in the knockout mice are so thick, it's hard to image. So we're still working on the exact quantification of the oxygen.
And you're studying with age-dependent mice, right? What about gender related?
Yeah.
There are differences.
So yes, we've been examining to see if there's any differences between male and female mice. And we haven't seen really much difference in the in our knockout mice, but I think the sample size is still too small. One of my grad students said she saw some differences in some of the inflammatory cytokines in the female mice, that they were a little bit higher in the females compared to male SOS knockout mice, but we have to do more studies on that. Well, we don't know yet. Yeah.
And the last one is, what about the extracellular matrix that is involved in the bone niche? Have you studied some cells?
I have not studied the extracellular matrix on the outside in detail. We know that the extracellular matrix is important for capturing some of the growth factors like CXCL12 and as they're secreted, but we haven't examined it to see if if the extracellular matrix is changed in the knockout mice. But that's an interesting question. Thank you. No, there's more. There's two more on there, so we have time.
Sí. José Andrés. José Andrés.
Hay una pregunta también en el Zoom.
Oh. She's sí.
For questions.
La pregunta es la siguiente. Dice, habla Manuel Durán de la Universidad UNISARC, último grado en curso. ¿Qué efectos tiene el cortisol en roedores con respecto al calcio? ¿Es quelante del calcio o no a la hora de diagnosticar osteoporosis?
So the question is, what is the effect of cortisol on the calcium, and if it's increased?
Diagnosis.
of osteoporosis. I don't know much about cortisol at all, but I can help him look for the answer in the literature if he wants to look. I don't know. Thank you for your question.
Y allá atrás hay dos.
Wow.
Ah, bueno. Dos allí, dos atrás.
Hi.
Hi.
Mateo, with my friend Thomas, we have a question. We saw that in the graphics, the B cells were like where the B cells are have more influence. The bone is not the percentage of bone is not in abundance. Like, for example, if we have so much B cells, we don't have a good amount of bone.
Mm-hmm.
And if we don't have B cells, we have more bone. Is that correct?
So I found that when you have the opposite, I found when you have more bone, we have less B cells. But I don't know if you have less bone, if you get more B cells. We have not found that.
No. It's for example, if we have more presence of B cells, our bones will be like
Thin. No, I don't think so. So we looked at the in the normal mice that have normal B cells, they have normal bone density. And we looked at mice that have overexpressed sclerostin, so not knocked out, and those mice have normal bone density. So I'm not sure if the mechanism of, besides the reduced growth factors for the B cells. I think the skeletal cells in the SOS knockout mice and the VHL knockout mice have some dysregulation and dysfunction, and that is making them affect the B cells. Contributing to the bone. There's some old papers that say that you need B cells to improve the bone density, but it hasn't been consistently found in other studies. So there's some evidence for what you think. Thank you for your question. Yeah.
Hello, my name is Ilona, and I have a little question. So first of all, let me know if I got this right. You're saying that if someone that has osteoporosis like takes NET for the osteoporosis, sorry. It's gonna the NETs are gonna take the path of the HIF, right? And then eventually, it's gonna affect the B cells, decreasing the amount of B cells, right? So maybe I was just wondering if you think that this side effect of the osteoporosis drugs are worse than the yeah, you got me right.
Yes.
The osteoporosis itself.
Yeah. So the question is, so what? Right. So we have fewer B cells, but are the animals getting sicker? Can they fight infection? We're still testing that. So we want to take I need to get new funding for that. So we want to infect the mice with different pathogens like bacteria or maybe viruses, and then see if they can respond as well as the untreated or the uninfected mice. So that's something in our minds.
Okay, so it's something that is still on research, right?
Yes.
Thank you.
Yeah, thank you for your question.
And I don't know, I want to know if you think there might be similar results when you, I mean, not in patients with osteoporosis or treatment, but in patients that have damage in their bones from things like trauma.
Oh, okay. So the question is there after trauma like broken bones or, okay.
Patients, they grow their teeth and how they generally climb in their pelvis. If there may be a correlation and have similar results in their immune system.
So it's interesting. The first step of healing bone fractures is inflammation. You need inflammation to start the healing process of the bone. I'm not sure if that process is slower in aged or elderly patients compared to younger patients. There have been studies. They're starting to do clinical trials now, looking after fracture on the sclerostin antibody treatment after fracture to help improve that. But I think the results are still being collected. They haven't been reported yet. So we'll see. Yeah, thank you. Okay, so son mis investigaciones bacanos. Gracias.
Felicitaciones. Felicitaciones, doctora Jennifer. Stefan, sorry. Recuerda que tenemos una titulación en un convenio que se hace con UNISARC y UNISARC. Por favor, entenderse entonces